Hello, everyone. Good afternoon, and thank you for joining us. We are delighted to have all the scientists from academia, biotech, and pharma here today for this session. Let's wait for another 1 minute before we start. Yeah, so welcome again. This is an official release webinar for our Covalent Suite, major step forward in the covalent drug design. Over the past few years, there has been a surge in interest in targeted covalent inhibitors, and this release marks our efforts to make the covalent design as systematic, transparent, and uh, physics-based as the rest of our SILCS platform. And today we will show what that looks like. My name is Abhishek. I'm an application scientist here at SILCS-Bio. I'll be moderating today's session and presenting part of the slides. We are joined by Dr. Oliver Tao, who is also an application scientist here at SILCS-Bio. And then we have Dr. Wenbo Yu, research associate professor at University of Maryland, Baltimore, who works with Dr. Alex Mackrell, who is also here, one of our co-founders, a professor at University of Maryland, Baltimore, and chief scientific officer for SILCS-Bio. And also we have Dr. Alvin Gubench, one of the co-founders for SILCS-Bio. So let's get started. We start sharing this slide. So before we go there, let's do a quick poll while people are still waiting, just to see how many of you have used or known SILCS before or currently using. So go ahead and work on that while I share the slides.
So, Abhishek, will people be able to put questions into the chat as the...
Yes. So yeah, also that, yeah, we have the Q&A section where you can basically put in your questions if you have during the presentation is going on, and we can address those questions at the end of the seminar. Can you guys see the slide yet? Yeah.
Yes, I see it.
Oh, okay. Cool. Let me start. That's my team's not responding correctly. Can you see my? Yeah. Let's see. Wait, is it working? Is it working? Okay.
You're stepping through the slides. It's
Looks like my Teams has kind of hung here. Sure. Can you still see the slides?
why don't you close the poll?
Yeah, my Teams has kind of hung, so I'm kind of not able to rejoin that.
Well, while we're waiting, I could like to say that, we've developed the SILCS technology, actually, Olgan Gavunt and I, back in, as I probably 2008. And the idea was to sort of, simulate, small molecules, solutes in and around a protein, and use that as a method to probe where different types of functional groups have favorable or unfavorable interactions. And what we were able to do is just use basic molecular simulations to do comprehensive sampling of solutes in water in and around the protein, from which we got the probability distributions and then converted them to free energy distributions
Okay.
and so on.
Like,
And
okay,
that's been the foundation of the
let's
technology where we can use these maps for many different things, and
Welcome again to
including
the
the
Covalent Suite
covalent.
release webinar. Here is agenda for today. For those who are new to SILCS, we will briefly introduce the SILCS method. Then Dr. Wenbo Yu will present the SILCS-Covalent module, followed by validations and feedback. Oliver will then take you through the command line interface of the SILCS-Covalent module. And lastly, we will end the webinar with a Q&A session. At SILCS Bio, we develop and distribute scientific software for computer-aided drug design based on the patented SILCS technology. Mainly, the different SILCS-based modules are collated as small molecule suite and biologic suite. Additionally, we provide single-step FEP and CGNFF suite that allows different options to use computational approaches in structure-based drug design campaigns. Let's begin with an introduction to SILCS. The fundamental idea behind SILCS, which stands for Site Identification by Ligand Competitive Saturation, is to use simple chemical solutes to map the functional group affinity of the entire protein. The probe molecules, along with water molecules, compete with each other to occupy the most favorable interaction sites on the protein surface. We extract this information, analyze, and process it to generate a pre-computed ensemble of affinity maps, which we call frag maps. We will show how this one pre-computed ensemble can then be used for various tasks in drug design campaign, such as site identification, virtual screening, hit identification, hit to lead, and lead optimization. Here is what undergoes in a silk simulation. We start with a protein and immerse it in a box with SILCS probes. Note that there is no ligand present in this case. 10 systems of the solvated target protein with scrambled side chains are generated as starting structures for 10 independent runs. We then run each of the 10 systems for 100 nanoseconds of MD simulation. After 1 nanosecond of MD simulation, we introduce an enhanced Grand Canonical Monte Carlo, that is GCMC simulation, for 100K steps before resuming the rest next MD simulation. Here, the MD simulations allow for conformational sampling of the protein as well as the probes and waters. And the GCMC simulation allows for sampling of probes in the deep pockets. The information from this huge calculation is then processed to generate the frag maps as shown on the right hand side here. To do that, we divide the full 3D space into tiny cubic voxels and calculate the probability of each probe occupying the voxel. Now here is a representation of what we find in the presence of a protein. The probability of the probe molecule occupying a given voxel is driven by its favorable and unfavorable interactions with the protein. There are regions with higher and lower probabilities than the probability of probes interacting with each other in the absence of the protein. And importantly, there are regions with no probability. All these probabilities are then normalized with the bulk solution probabilities and Boltzmann transformed to get a grid free energy at each cubic voxel. This is done for all types of atoms in the set of probe molecules, giving us the final frag maps. Now, We can visualize these frag maps in reference to our starting structure, visualizing different frag maps that represent apolar, hydrogen bond donor, hydrogen bond acceptor. Positively and negatively charged regions can be quickly analyzed for qualitative assessment of your target. Additionally, different GFE levels allow for regions that will bind the specific chemical class most tightly to be identified. Unfavorable regions may also be identified. Now, to use frag maps in a quantitative way, we first take the ligand pose and overlay in the field of frag maps. We then take each ligand atom and assign a frag map type to it. Then, based on the location of the atom and the grid free energy at that location, we can then get an atomic contribution to the ligand's energetic favorability. Basically, we can get an atom based free energy contribution that can be summed into a score that we call LGFE, ligand grid free energy. This LGFE score is an estimate of binding free energy of the ligand in that pose. Because the frag maps are pre-computed, this simple assignment and summation is instantaneous and becomes the core of this technology as we move forward. The SILCS technology is not limited to overlaying ligands onto frag maps. We can also subject the ligand to the conformational sampling using what we call SILCS Monte Carlo or SILCS MC docking. In MC docking, we sample the translational, rotational, and torsional degrees of freedom of the ligand in the field of frag maps. The Metropolis criteria uses intramolecular energy of the ligand conformation and the LGFE to optimize the function of finding the best ligand pose and most favorable LGFE score. We can do this in a full docking mode where we are sampling the full rotational and translational space of the ligand in, say, a binding pocket, or in a mode we call pose refinement, which is more local relaxation from a known starting conformation, such as from a core crystal structure. The full docking takes typically an order of approximately 1 minute or so, so representing a speed up of over 1,000-fold compared to traditional free energy calculations. Here are results from a recent validation study that compares the free energy perturbation methodology with SILCS method. We have presented a comprehensive study of approximately 200 ligands across 8 different targets where we compared SILCS with number of previously published FEP studies. It is very evident that LGFE scores provide highly comparable relative binding affinity to the free energy perturbation calculated delta G values. And when using iteratively with experimental data, and this experimental data can be for small number of compounds as small as 10 molecules, we were able to improve the relative ranking of the ligand affinities shown by the percent correct data here on the right column, where for standard silks the percent correct was 0.71. With silks MC machine learning optimized approach, we can get it up to 0.81, which is on par and above the standard free energy perturbation methods that are out there. To summarize the key advantages of SILCS bio approach, it is the pre-computed GFE frag maps, allowing rapid and accurate ligand binding affinity estimates in a way that leads drug design process. And beyond the quantitative predictions, usually the frag maps easily allow for an understanding of types of functional groups that can be added to a molecule to increase its binding affinity, which is very useful in the medicinal chemistry to give ideas about synthetically accessible molecules. We note that frag maps explicitly count for protein flexibility. This is important because we know the protein can undergo local conformational changes in presence of different ligands. The frag maps account for desolvation effects, and this refers to the normalization procedure, which we described earlier. In addition to globular proteins, We also support membrane proteins such as GPCRs, ion channels, and transporters. We also have a specifically optimized protocol to apply the same method to RNA targets.
Leveraging the success of the SILCS technology as introduced by Dr. Abhishek Koganul, we further developed its functionality to address covalent drug design applications. I'm going to go through some details about this SILCS-based workflow for targeted covalent inhibitor design. Traditionally, drugs are designed to interact non-covalently with biological targets. But covalent drugs enhance this by forming irreversible or hardly reversible covalent bond with its target, a concept in use since aspirin's development in the late 19th century. Targeted covalent inhibitors, or TCI, combine covalent and non-covalent interactions by using reactive warheads and optimize the scaffolds to achieve greater potency, selectivity, and duration of action than traditional non-covalent drugs. Till now, there are multiple FDA-approved TCI drugs on the market. Computer-aided drug design is again an essential tool in covalent drug design, and is usually formulated by combining traditional docking methods with specialized modeling to account for covalent bond formation. Unlike non-covalent docking, covalent docking must account for local conformational changes in the target protein due to the covalent bond formation, which means the consideration of conformational flexibility is essential. Our SILCS simulation already holds the information about the protein residue samplings and thus can be conveniently leveraged for covalent drug design purposes. In the SILCS simulation, conformational space of protein residues is well sampled. In case of TCI development targeting cysteine, for example, to capture the spatial flexibility of the cysteine seal sulfur, 3D probability map can be generated with voxels occupancies normalized to reflect the likelihood of sulfur presence on a 0 to 1 scale. And this is so-called PROT map, or protein probability map. Together, these frag maps can be used to facilitate the TCI design with full consideration of target conformational flexibility. Using the information content in the frag maps and PROT maps, along with the implementation of machine learning modeling, the SILCS-Covalent module was developed to cover the 3 stages of TCI design, including identification of potentially reactive nucleophilic residues, selection of optimal warhead for specific residue, and optimization of the non-covalent scaffold to improve binding and specificity. In the SILCS-Covalent workflow, we use SILCS-MC to dock electrophilic warhead fragments as well as TCI molecules to produce matrix suited for a range of application scenarios. For selected nucleophilic residues such as cysteine, lysine, arginine, or serine, we have conformational samplings of the reactive item in the residue represented by protomap ready. Then we run exhaustive SILCS-MC docking of warhead fragment or TCI molecule around the reactive item of residue to sample their interaction conformations around the nucleophilic residue. The exhaustive SILCS-MC simulations are initialized from molecule conformations randomized in 6 spheres centered at 6 positions located 3.5 angstrom away from the reactive item in the 6 directions along the 3 axes. A simulation radius of 6 angstrom is adopted for the warhead fragment, considering their relative small sizes, while a 10 angstrom radius is used for the ligands. This protocol ensures we get sufficient number of samplings of binding poses around the nucleophilic residue. From here on, we're going to focus on cysteine as the nucleophilic residue, but our protocol can be easily applied to other nucleophilic residue cases. In addition to the regularly used metric ligand-grade free energy, which is used to measure the binding strength, here we define another metric to account for the probability of nucleophilic residue contact with ligand, called prob-res. It measures the likelihood of a molecule's reactive atom overlapping with the target nucleophilic residue protomap in favorable binding poses by integrating both geometric alignment and energetic compatibility for covalent bond formation consideration. For each cysteine sulfur probability voxel with a finite value, its distance to the Wall-height group reactive atom in a docking pose with favorable LGFE is measured. Summation is performed over all the recorded probability voxel values to get a contact probability for each docking pose. A prob-res value of 1 for docking pose means all non-zero sulfur probability voxels are within the user-defined cutoff distance to the Wall-height group reactive atom in the docking pose. The cutoff is used at here to define the maximum distance at which electrophilic atom to the cysteine sulfur will have the potential to react. In our SILCS-Covalent workflow, we model the TCA binding as a two-step process. A covalent ligand will first bind with the target protein non-covalently, which can be defined by the experimental K-on and K-off, from which a reversible equilibrium constant and free energy of binding may be obtained. The LGFE equilibrium or LGFE EQ from the SILCS-MC, which represents the ligand's pose with the most favorable LGFE, is relevant to this binding event. The second step towards covalent bond formation may be defined by the overall experimental rate constant K in NECT, which may be modeled as two consecutive processes. For the reaction to occur, the ligand needs to first adjust its orientation to position its Wall-height near the nucleophilic residue. Such orientation is defined as reaction-competent binding conformation and is identified based on the overlap of the Wall-height reactive functional group with the cysteine sulfur probability distribution in terms of prob-res. And the final stage of covalent bond formation can be quantified by the intrinsic reactivity of the Wall-height, which can be measured experimentally. For example, for the cysteine case, the glutathione reactivity rate constant, KGSH, can be used. As a summary, the metrics used in the TCA modeling are LGFE-EQ for the equilibrium binding state, the LGFE-Rx for the reaction-competent state, and the contact probability, prob-res. The first application scenario of using metrics that are derived from SILCS-MC is to identify reactive nucleophilic residue in a target. A Wall-height library that contains various typical Wall-height fragments is used for exhaustive SILCS-MC docking around each nucleophilic residue in a target to evaluate their interaction preferences. For example, for the protein as shown at here, docking of a Wall-height library is done toward all three cysteine residues. In the current setup, we are using Petrie's Wall-height library for targeting cysteine. Other Wall-height libraries could be used for other purposes. This library contains 24 covalent probe molecules based on a non-covalent scaffold and covers a variety of representative electrophils, including acryl, nitro, haloacetamide, and acetylene. After running exhaustive SILCS-MC docking of all Wall-height fragments targeting all nucleophilic residues in a target, the best probe res per wall height per residue is collected and are averaged over all tested wall heights for each residue. This metric is used to find tractable cysteine residues, since a larger averaged probe res value for a cysteine residue indicates that the residue samples conformations accessible to a broad range of wall height types, thereby potentially serving as a reliable nucleophilic residue that could be targeted by TCI. And in the current example, we can see the cysteine 481 is the one with the highest averaged probe res. We validated this metric using several protein test cases, and the residues with high averaged probe res value are mostly often to be found as the well-known targetable residue from literatures. For example, for the BTK protein, the cysteine residue C481, which has the highest averaged probe res value, is actually the well-known cysteine residue many FDA-approved drugs are targeting. Beyond the known targetable residues, this metric may also offer insights to other unidentified tractable residues. For example, for GST01 protein, C32 is a known tractable residue for TCI. There is another cysteine residue, C112, which also shows high averaged probe res value that could be targetable. Actually, this residue is also predicted to be targetable by other computational studies. After a nucleophilic residue is predicted, wall height groups with the highest potential to react with this specific residue can be identified and suggested for chemists to design TCI. To facilitate this step, metrics from SILCS-MC as well as experimental measured intrinsic reactivity are used as inputs to train random forest machine learning model using the activity data from Patrick's work, considering the 24 wall heights targeting the 6 kinases. When there is new data target new protein is available, we can easily retrain the machine learning model for better performances. And the machine learning model can inform which wall height group is the optimal and has the high activity potential to target the specific nucleophilic residue, which offers the chemist useful information for the initial step of TCA design. Once we have a wall height group ready, we come to the stage to further optimize the non-covalent scaffold of the TCI. And we can look at the docking poses of a TCI using SILCS-MC. The binding pose with the best LGFE, which represents for the equilibrium binding state, can be checked first. As a showcase, a known BTK TCI is shown at here. So as seen in the figures, even though the non-covalent scaffold in the equilibrium docking pose is aligned well with the crystal binding pose, the wall height group is not necessarily aligned, since the ligand will first non-covalently interact with the target protein. Next, the reaction competent docking pose, which has the best contact probe res value with the cysteine residue, can be further checked. This time, you can see there are good alignments between the crystal pose after bond formation with our reaction competent docking pose, considering both the non-covalent part and the wall height part of the molecule. In practice, one needs to balance the equilibrium and the reaction competent state to allow a balance between binding affinities at different binding stages, which could satisfy both affinity and reactivity requirements for an optimal TCI design. And both docking poses and related LGFE docking scores, as well as the atomic level GFE values, could offer abundant information to facilitate such TCI non-covalent scaffold optimization. So as a summary, we just went through the main capabilities of the SILCS-Covalent workflow. I am going to hand it over to Dr. Olive Tao for a further demo.
Let's go through the brief demo of SILCS-Covalent. If you haven't configured a SILCS-Bio environment, please check our user guide at docs.silksbio.com and follow the setup steps. If you need a trial license or help, interface. To run SILCS-Covalent, you need to call this command with your protein PDB file and pre-computed frag maps. You can choose warhead or ligand for the docking mode, depends on your ligand type. We provide Petri's warhead library for you to try. Currently, we support cysteine, lysine, serine, and threonine for the reactive residue. You can choose specific residue ID for covalent link or skip certain residue ID. Once you start the SILCS-Covalent job, it will submit many jobs to your job scheduler. You can check out the SILCS-Covalent publication for more details. Once the SILCS-Covalent job is finished, you need to run the second step to collect the docking results. In this step, you need to specify the protein PDB file, the result folder from the first step, and the warhead ID file. The warhead ID file should contain two columns, the name of the compound, whether it's warhead or ligand, and the reactive atom ID. You can modify the distance cutoff for covalent bond, the probability of the reactive atom, the ligand efficiency, or LE cutoff. By default, this job runs on the head node. You can use option batch equals true to submit the job to your queuing system. After collecting all the warhead docking results, you can check the avg underscore progress-rx.dat, which is for the average probability of reactive residues. In the BTK example, we used Petri's warhead library to dock on all cysteine residues. In the data file or this chart, we can see the cysteine residue 481 has the highest average probability, which makes sense because the current FDA-approved BTK drugs are targeting this particular residue. Last but not least, in the BTK example, we used SILCS-Covalent for redocking the crystal structure. The result CSV file shows both LGFE score and the progress for EQ, RX, and the RxEQ. All the docked poses are saved in SDF format by default. You can load the docked pose to your preferred visualizer and compare it with the crystal structure. In this case, SILCS-Covalent did a pretty good job for redocking the crystal ligand.
Thanks, Wenbo, for presenting the SILCS-Covalent approach, and thank you, Oliver, for showing how one can run these calculations with two simple steps. This brings us to the Covalent suite of SILCS-based modules. So today we are excited to present the Covalent Suite as a collection of the basic modules from the small molecule suite with an addition of the SILCS-Covalent module. This allows the users focused on covalent drug design to access the most relevant SILCS-based modules.
Thank you everyone for tuning in. We are excited to bring you the Covalent Suite. For more details on our new development, please visit us at docs.silksbio.com. And for general inquiries, please visit us at silcsbio.com or email us at info@silcsbio.com. We are happy to take any questions.
All right. Let's see if we have any questions. Put in the chat. People can use the Q&A section to send in any of their questions in the meantime while we go through the questions. Okay. Does the Covalent Suite include example input files or tutorials to help new users get started? Yeah, so we will be providing the proteins that were used in the validations along with the Petrie's results in the suite that users can access and validate the the results again if they want to. Yeah. Okay. Is the probabilistic prot map generation influenced by simulation length or starting structure? And how sensitive are progress results to that? Wenbo, you want to answer that?
Sure, I can take that question. So similar to other SILCS applications, the starting structure matters because the conformational flexibility being considered in a microsecond MD simulation it's kind of limited to the local conformational changes. And if you're looking at a protein with large conformational changes and have multiple conformational states, then each of the conformational states could be used to start a different silk simulation in order to get a relevant collective of conformational ensembles to get the following silk applications to produce meaningful data. So yeah, so the structure like matters.
Right. There is one question in chat. Thank you for putting this together. Says Valentina, if we were to target a different reactive residue, different than cysteine, are there guidelines to build the required warhead library? So currently we are supporting lysine, serine, and threonine along with the cysteine. So we can provide the guidelines to build the warhead libraries if you know which warheads you are bringing in. Basically, you need to create an STF file of all the warheads and then create a warhead ID file that will list all the reactive atom IDs so that we can do the progress analysis accordingly. So yeah, we can provide the guidelines for sure in building that required library. Okay, other question. In systems with multiple reactive residues in proximity, how does the workflow prevent overlap or false positives in progress calculations? Wenbo, you want to take that?
Sure. So our protomap for electrophilic residues they are generated separately for each residue. So the sampling of the warheads or TCI like targeting each residue will be only focusing on that residue without the map contamination like from nearby residues.
Yes, yeah. So while you're generating the frag maps, we have the option to provide, a res ID list so that you can create that individual residue protein map that allows us to, get away from that problems of overlapped protein maps. Yes. If and how can users retrain or expand the ML warhead prioritization model with their own internal datasets? Yeah, so basically at this point, we have the machine learning model to trained on the data from the Petrie's warhead database for specifically for the cysteines. And we have the Python modules that uh, build that machine learning model uh, available for users to basically change based on their own data set that they are bringing in. You'll have to bring in your own experimental data and the full warhead library for which you will run the same calculations on different known targets. And then you can rebuild the machine learning model and use that in your inference for new data. Anything there, Wenbo, to add?
Yeah, that's pretty much right. So if user has their own data set, like it will be very helpful to further enhance the machine model's predictability. So they can just use our SILCS-Covalent workflow to get all the metrics and then use them as input features to easily retrain their own machine learning model. We have all the scripts to facilitate the automation of such like machine learning retraining process. Yeah.
Right. Okay. Do you anticipate combining SILCS-Covalent with AI-driven generative design to propose covalent ligands automatically? I will let Alex take that question.
Yeah, that's certainly a possibility. At this point, we haven't started to do those types of developments themselves. However, what's nice about the SILCS technology is that you could use other AI generative tools to create possibilities and then rapidly evaluate them using the SILCS-Covalent module and so on. So it could be directly linked to such tools. We are not developing that technology right now ourselves.
Cool. Thank you. Bora has a question. I also have a question about UI. At the moment, as far as understood, it runs from the terminal. So do you have any plans for creating an UI and make the workflow easier for people getting into this field? Yes, definitely we have been porting all our command line interface tools into the graphical user interface that we have right now. So in the future, we definitely plan to incorporate the SILCS-Covalent approach into the graphical user interface. Thanks. Another question, do covalent molecules have any size limitations? Can we use small polymer? Also, is Covalent Suite computationally expensive? Take that, Alex.
Yeah, I mean, yeah, you can do the SILCS Monte Carlo docking is very flexible, and you can do larger molecules, smaller molecules. If you're doing larger molecules, you may want to increase the amount of sampling of dihedral degrees of freedom and so on. So yes, I wouldn't do a polymer that's too long, but you can use sort of a few subunits and do the docking with that and so on. Yeah, that shouldn't be a problem. Is the second part of the question?
Yeah, the second part was just, is Covalent Suite computationally expensive?
Again, with the SILCS technology, you pay your upfront cost for the SILCS simulation, which may be for a kinase, a couple days on 10 GPUs. But once you've got those frag maps and the protein maps available, the SILCS calculations of the individual ligands are fast, again, in the order of minutes each. So it's very computationally efficient and so on. And again, those same frag maps can be used for different parts of the SILCS suite of capabilities, just general MC docking for protein-protein interactions, for a variety of different SILCS methodologies, all use those same set of frag maps. So again, we do that one set of silk simulations that may take a couple days, larger for like a GPCR, more for GPCR. But that pre-compute ensemble of frag maps, you can use them over and over again, and they're very computationally efficient.
All right. And there's a question about force field. So in the MD simulation, which force field is used? And please tell me, does this simulation is possible in SILCS-Bio, as it may be little difficult to perform it in externally. Yeah, currently we are using the CHAMP 36 force field for the proteins and the CGEN-FF force field for small molecules. You probably will not be able to run the same simulation outside, because we have validated it against different measures over the years to include that set of six solutes that we have. And also the grand canonical Monte Carlo sampling is highly validated and tailored over the years. So you will have to get the license to run the calculations. Okay, I think there is some question in the
Yeah,
chat.
you could run them internally. I mean, SILCS-Bio can run, silk simulations for customers internally.
Okay, there is a question about, is it necessary to have an initial idea of possible covalent interactions between the ligand and the protein I'm testing, or is it possible to screen?
The whole SILCS covalent workflow is designed to start from no knowledge at all. Okay. So you go in, you do the standard silk simulation protocol, and then you go through the three steps of ranking the different cysteines or lysines or whatever residue you're targeting, and then following up with additional analysis and so on. So no, you don't have to have a preconception about which residues you can target and so on. This is sort of one of the powers of the technology.
Right. Anything else in the Q&A? Okay. Yeah. How do the PROGRESS and LGFE metrics compare to traditional covalent docking scoring functions like in CoDock and Gold? Wenbo, do you want to take?
Let me take that question. So till now, like we haven't done any comparison tests with other covalent docking methods on the market yet, but it could be done in the near future. But I want to mention the differences between our protocol with other on the market protocols. So our protocol use this embarrassed and reaction competent docking. So it holds the computational simplicity of the method that can be used both for a covalent drug design or non-covalent drug design. While CoDock or Gold, they conduct a post-reactive docking. So they have specialized treatment on the covalent bond formation part. While they are more focusing on how a ligand could be accommodated in the binding site after the reaction, we are focusing on the important binding steps of a TCI, which can be directly linked to the experimentally measured K on K off as well as the K inject. So if we also take the intrinsic reactivity into account, so that's a major difference.
Yeah, I'd like to also
yeah.
say, in the silks, the information content of the, you have the overall LGFE scores for the equilibrium and the reactive complexes, but then you can partition those LGFE scores into the energetic contribution of different portions of the scaffolding regions and so on. And this is information on individual moieties, what they're contributing to the different bound conformations. And that's information that can directly form ligand design. So that's the type of information on, is this hydroxyl contributing a lot? If I change that hydroxyl to a methyl, how does that contribution change? Does it affect the contribution of other parts of the molecule? So you have this sort of atomic level free energy granular information that greatly facilitates ligand optimization. And that information is just not accessible to the other methodologies.
All right. We have one more question, I think, in the chat. Can the SILCS Bio possibly run MD simulations on proteins with iconic bonds? Maybe they want to say ionic bonds. Yeah, that's, I think it's not possible.
Yeah, if it is, I would assume it is ionic bonds. So
yeah,
yes, I mean, you're using the standard charm force field, which you can treat all sorts of different protonation states. You can even build in different types of non-natural amino acid residues. It's very flexible. The capabilities are quite broad with respect to, the properties of the protein and so on. So
yeah. I want to add to the previous question about like restraint docking, comparison with others. So we have the restrained MC docking that can be done in terms of like if you know where the warhead ID is and where the the reactive atom of the residue is, then you can put a restraint on the one of the atoms and then run this restraint docking in the SILCS-MC docking. Just posted the link to that part. So that can be used in post reaction kind of evaluations, right? That Wenbo mentioned. When are another question? When are covalent inhibitors considered therapeutically? Okay, that's
all the time. I mean, it's very common these
exactly.
days. It's just, for many years, it was like, as Wenbo showed a timeline of, covalent inhibitors have been around for a long time. For a number of years, people were sort of stayed away from them, but now they've become much more of much wider interest, and they're used for a wide variety of proteins. It's just, do you have the appropriate nucleophile, cysteine, lysine in a region of the protein that is related to activity? So covalent inhibitors are widely considered, and many have been approved by the FDA and so on.
All right. Yep. Another question from Sivasakti. Since covalent warheads may have intrinsic toxicity or reactivity, does the software provide any integrated ADME tox filters, or is experimental validation always necessary to rule out safety issues? Yeah, we don't have the ADME or tox filters internally integrated in the approach. The workflow is providing you the ideas about what kinds of residues are most potential reactive targets, and then you have the warheads that you are bringing in to analyze. So any experimental validation, you will have to bring that in separately about the safety issues or ADME tox filters. All right, that's about it. Thank you everyone for again joining in. We will be sending out the recording of the webinar in a few days or so once this is done. So you can share it with any of your colleagues that couldn't make the webinar or anything. And yeah, please visit our website and send us any questions if you have. We're happy to answer them. Thanks, Wenbo. Thanks, Alex. Thanks everyone for joining. See you next time.
Take care.